Good morning. Today we are to discuss about the role of bacterial plaque and its stages of formation, its role in dental caries. Plaque is a tenaciously, strongly adherent deposit that forms on tooth surfaces. It consists of an organic matrix. The organic matrix is composed of a dense concentration of bacteria. In microbiological terminology, plaque is a biofilm of bacteria that are embedded in an extracellular polysaccharide matrix. In the protected environment in a biofilm, conditions are very different from those on a clean tooth surface or in saliva. Bacteria inside the biofilm can exhibit cooperative activity and behave differently from the same species in isolation in a laboratory culture medium. It means the bacteria in plaque will behave differently than it behaves in a culture medium in the lab. The property may confer resistance to antimicrobials or to immunological defenses to which the individual bacterial species are normally sensitive. The biofilm traps and concentrates bacterial products, favoring survival and interactions between species. Therefore, bacterial plaque must be regarded as a living ecosystem and not as a mere collection of bacteria. A very important key feature is the ability of dental plaque to concentrate and retain acid. When we talk with clinical perspective, bacterial plaque is a tenaciously adherent deposit on the teeth. It resists the friction of food during mastication and can only be readily removed by toothbrushing. However, neither toothbrushing nor fibrous food will remove plaque from inaccessible surfaces or pits stagnation areas. This tenacious adherence is mediated by the matrix polysaccharides. Plaque becomes visible particularly on the labial surface of the incisors when toothbrushing is stopped for 12 to 24 hours. It appears as a translucent film with a matte surface that dulls the otherwise smooth and shiny enamel. It can be made visible when stained with disclosing agents. Under conditions of starvation, little plaque forms, but it forms rapidly and abundantly on a high sucrose diet. In stagnation areas where plaque is undisturbed, growth of bacteria and secretions of matrix thicken the plaque. If sugars are available, they are metabolized to the acid that causes dental caries. Now we will talk about the stages of plaque formation. If the teeth are thoroughly cleaned by polishing with an abrasive, plaque quickly reforms in following sequence. First, there will be deposition of structureless cell-free pellicle of salivary glycoprotein. Then there will be further deposition of pellicle enhanced by bacterial action precipitating salivary proteins. It will be followed by colonization of the cell-free layer by bacteria, particularly by Streptococcus sanguis and Streptococcus mutans strains within 24 hours. There will be progressive buildup of plaque substance by polysaccharides produced by bacteria. In last, proliferation of filamentous and other bacteria takes place as the plaque matures.